Hello friends, welcome to today's session of Biochemistry. I am Dr. Nilesh Chandra and we are going to continue from carbohydrates where we stopped yesterday. Alright, so just give me send your hi, hello so that I know that you're here. I cannot yet see students. Come on, log in and send me your hellos. How many people have joined in till now? Can we have a head count? Please send in your attendance. Please send in your attendance. Hello friends, welcome. Hello Sujan, waiting for more of your friends. उड़द तो बहुत खाए दिन खाए रहा न जाए चूहा दिन में कैसे जाए हाँ? Great. Okay, so I hope the audio is clear. I hope the video is clear. Can you all hear me? Am I audible across to everyone? Can you hear me? I think the video has to be a little bit. Let me see. Okay, better. Mm, yeah. Can you hear me? Can you hear me? Am I audible to everyone? Am I audible to everyone? Just send your comments for the voice, send your comments for the video, and I hope there is no lag today. Is the audio clear? Is the video clear? Good. Okay, so this is where we had stopped yesterday. We were talking about the glucose transporters yesterday, and today we'll be talking about the HMP shunt. We'll talk a little bit about the glycogen metabolism. We'll look at the glycogen storage disorders, and we'll look at the galactose and fructose metabolism as well. All right, but before we go there, let's see few questions for you. I would be expecting you to answer them quick fire. Look at the question, send in your answers. Glycolytic enzyme inhibited by a fluoride. Your options are glucokinase, hexokinase, enolase, and aldolase. Which of them is the correct answer? Is it stuck? Very good. The correct answer is enolase. The correct answer, as you know, is enolase. Enolase is inhibited by a fluoride. Commonly, the fluoride would be available as the sodium fluoride. So the correct answer is definitely enolase. Very good, very good. Well done. The correct answer is enolase. Let's move on to the next question. Why is the video not streaming? Let's move on to the next question. Why is the video not streaming? Mac. What happened to the video capture device? Hello? Hello. Display capture, video capture device. What happened to the video? Remove. Okay. Add video capture device. Create new. Okay. Okay. There we go. Okay, that should be it, I hope. Okay, so yeah, I know it was stuck, so I'm resetting it. Visual got stuck. I know. Okay, so 3 ATP in anaerobic glycolysis occurs with which fuel? 3 ATP in anaerobic glycolysis occurs with which fuel? We had done this. We had done this question, and the correct answer is glycogen in the muscle. Glycogen in muscle is the correct answer. Glycogen in muscle is the correct answer. All right. So 3 ATP in anaerobic glycolysis occurs with glycogen in muscle. With glucose, fructose, galactose, you're going to get the 2 ATP. Anticoagulant used to estimate glucose from a sample sent from PHC is. Your options are sodium fluoride, EDTA, sodium citrate plus EDTA, and potassium oxalate plus sodium fluoride. So, which of them is the correct answer? The anticoagulant used to estimate glucose from a sample sent from PSC. See, typically, when a sample will be sent from PSC to the lab, it is going to take about 3 to 4 hours minimum to reach to the lab. Before that, it cannot reach there. So you have to keep that in mind before you give the answer. Sodium fluoride is the most common wrong answer. Sodium fluoride is the most common wrong answer. Please note, sodium fluoride is not correct. I told you sodium fluoride must be mixed with a strong anticoagulant. We have written this either potassium oxalate or sodium potassium EDTA in the gray vial that we have currently in the hospitals, the Vacutainer. We have the sodium potassium EDTA plus sodium fluoride. So the correct answer is option 4, not option 1. Option 1 is the most common wrong answer. Please note that is not the correct answer. Take a look at this question. After overnight fasting, levels of glucose transporters are reduced in which of the following cells? Your options are brain cells, hepatocytes, adipocytes, and RBC. After overnight fasting, level of glucose transporter are reduced in. What is your answer? Now, what you have to understand here, after overnight fasting, what will happen? The glucose concentration will fall. When the glucose concentration when the glucose concentration falls, there will be decrease in insulin concentration. Therefore, you have to look at the tissue where glucose transporter is dependent on insulin. Which glucose transporter requires insulin? And the answer in that case is GLUT4. And where is GLUT4 present? Adipocytes and muscles. So the correct answer is adipocytes and muscles. The correct answer is adipocytes and muscles. In adipocytes and muscles, our overnight fasting, because the level of glucose will decrease, the level of insulin will also decrease, and the GLUT4 transporters will be internalized. They will go inside, and therefore concentration on the surface will be decreased. Because they are internalized, the concentration on the surface will be greatly decreased. Okay, so let's go on to the next question. Pyruvate can be converted directly to all of the following except. Now, this is a very straightforward question. Which of the following cannot be formed directly from pyruvate? Your options are PEP. PEP stands for the phosphoenol pyruvate. It stands for phosphoenol pyruvate. Alanine, acetyl coenzyme A, and the lactate. So, which of the following cannot be converted directly from the pyruvate? So, we had seen the alanine can be obtained from ALT. Alanine transaminase will quickly convert the pyruvate to alanine. Acetyl coenzyme A can be obtained from PDH and lactate from LDH. So, obviously, the correct answer is phosphoenol pyruvate. We had seen two intermediate enzymes, the pyruvate carboxylase and the PEP carboxykinase, are required to convert the pyruvate to PEP. Direct conversion from pyruvate to PEP cannot occur. Direct conversion from pyruvate to PEP cannot occur. We don't have a corresponding enzyme. This is a slightly tricky question. It ensures whether you were attentive in the class or not when we were discussing a topic on PDH. Arsenite inhibits all except, your options are PDH, the pyruvate dehydrogenase, the lipoic acid, the branch chain keto acid dehydrogenase, and the isocitrate dehydrogenase. So, which of the following is the correct answer? Arsenite inhibits all of the following except. Now, here what you have to remember, the arsenite directly inhibits lipoic acid. The arsenite will directly inhibit lipoic acid. The lipoic acid is present in PDH and branch chain ketoside dehydrogenase. Remember, the PDH is similar to the branch chain ketoside dehydrogenase and the alpha ketoglutarate dehydrogenase. We had said two enzymes and five coenzymes are similar in the branch chain ketoside dehydrogenase, the alpha ketoglutarate dehydrogenase, and the pyruvate dehydrogenase. Only the first enzyme is different. Therefore, the PDH will also get inhibited. The branch chain ketoside dehydrogenase will also get inhibited. So the correct answer in this case, as most of you have written, is the isocitrate dehydrogenase. Arsenite inhibits all of the following except the isocitrate dehydrogenase. Very good. Let's go on to the next question. Congenital lactic acidosis may occur due to defect in which of the following enzymes? Now remember, congenital means from birth. This means we are looking at a genetic defect. We are looking at a genetic defect. Your options are pyruvate decarboxylase, the pyruvate dehydrogenase, the trans ketolase, and the succinate dehydrogenase. Which of the following will result in congenital lactic acidosis? Now, it's seen lactic acidosis is seen when PDH is not working. When the PDH is not working, that is when the lactic acidosis occur. And we had seen when there is inherited deficiency of PDH, that is when we see the congenital lactic acidosis. So the correct answer in this case is the PDH, inherited deficiency of PDH. Please note we don't have an enzyme of pyruvate decarboxylase. We have the pyruvate carboxylase but not the pyruvate decarboxylase. Independent enzyme pyruvate decarboxylase is not there. Okay, very good. So the correct answer, as most of you have correctly written, is the pyruvate dehydrogenase. Let's move on. In vivo control of citric acid cycle is affected by. When I say in vivo, what do I mean? In vivo means inside the body. In vitro means in the lab. In vivo means inside the body. So inside the body, control of citric acid cycle is affected by which of the following? Acetyl coenzyme A, ATP, and citrate. These are the options. So we had written the target, or you can say the regulatory enzyme. We written the regulatory enzyme was the isocitrate dehydrogenase. I'm sure you remember this. The regulatory enzyme was isocitrate dehydrogenase, and the isocitrate dehydrogenase is inhibited by ATP. Isocitrate dehydrogenase is inhibited by ATP. So, in vivo control of citric acid cycle is affected by ATP. The answer is ATP. We had seen that citrate is able to regulate the glycolysis, but is not able to regulate the citric acid cycle per se. It cannot directly regulate the citric acid cycle per se. So the correct answer in this case is the ATP, option number 3. And we have not yet discussed the biological oxidation, but I'm sure most of you can answer this question about oxidative phosphorylation. Which of the following statements is true? About oxidative phosphorylation, which of the following statements is true? I'm curious, how many of you are having munching something like me? Can you send us what you are munching so that we all can also enjoy the different dishes that all of you are having and the comfort of your home? What food are you eating? What drink are you having in your room while you are having the class? Can you send the message? You would like to enjoy the same. The answer to this question is generation of ATP. Generation of ATP. The oxidative part is utilization of NADH. The phosphorylation part is generation of ATP. So until and unless ATP is generated, the whole process is not completed. Utilization of NADH is only oxidation. Utilization of ATP is NADH is only oxidation. It is not oxidative phosphorylation. Snack at 10 p.m. Coffee. Okay. What else? If you have snack at 10 p.m., when are you going to have your dinner? Srujan, when will you have dinner? Maggie, papaya. Okay. So see, that is the another benefit of having the online class. We can enjoy so many food simultaneously. Dinner after class. Okay. 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 Shall. So let's go back. Let's go back. So remember, the utilization of NADH is only the oxidation part. The phosphorylation will happen once the ATP is generated. So when I say oxidative phosphorylation, it is a two-step process: oxidation followed by phosphorylation. Utilization of NADH followed by generation of ATP. So the best answer here should be generation of ATP. Generation of ATP should be the best answer. Good. Another very simple and straightforward question. Everybody knows this. The cytochrome C mediates, and your options are electron transport, uh, glycolysis, uh, necrosis, and apoptosis. Okay, me. Uh, I'm having a bit of noodles, but this is not Maggie. This is homemade noodles. So because nowadays I'm on night duty, I 2 days back we invited a little kid in my house. So nowadays I'm on night duty. So I have to fortify myself for the night duty. Okay. So let's come back to the question. Cytochrome C mediates. Very good. Very good. All of you have given the answer as electron transport. Very good. Electron transport. Murtaza has given us the answer as apoptosis. And Dr. Manohar has also given us the answer as apoptosis. Please note, electron transport occurs inside the mitochondria. The question says cytosolic cytochrome C. See, this is a question having only 4 words, out of which you have ignored the first word. You have not read the first word, you have just read cytochrome C mediates. So, I just want to emphasize, when you go for the exam, make sure you are not hypoglycemic so that you are able to concentrate. When you're sitting in the class, make sure you're not hypoglycemic, you are able to concentrate, and read the complete question before answering. Read all the options before answering. If you don't read the complete question, if you don't read all the options, it is very likely that you will make a mistake. All right. The cytosolic cytochrome C is the activator of apoptotic cascade. It is the activator of apoptotic cascade. So please note this point. Please note this point. The cytosolic cytochrome C is activated apoptotic cascade. The cytochrome C will come out into the cytoplasm only when there is damage to the membrane permeability of the inner mitochondrial membrane. Until unless it is damaged, the cytochrome C cannot come out. A damage to the inner mitochondrial membrane indicates impending apoptosis. That is the first finding in the apoptotic cascade. So remember, cytochrome C in the mitochondria is part of electron transport chain in the cytoplasm. It is the activator of apoptotic cascade. It will result in apoptosis. So be very careful when you are answering the questions. Now this question we had done right in the beginning on day one, when we are doing the classification of the carbohydrates. At that time we had discussed the reducing substance in urine is seen in all except. Reducing substance in urine is seen in all except, and your options are lactose intolerance, the phenylketonuria written as PKU, the galactosemia, and the Fanconi's anemia. A lot of time it will happen that you may not know the uh, different answers in context to the question. Various options are mentioned, you may not know all the options, but sometimes by knowing a single option you are able to arrive at the correct answer. Can anyone tell me the answer to this question? Very good. Very good. Remember, we had mentioned the reducing substance in lactose intolerance is there in stool. It is not absorbed, so no reducing substance in the urine. So this is the correct answer. This also means the reducing substance is present in PKU, in galactosemia, in pancreas anemia, in the urine. Please keep this in mind. There is an additional information. Reducing substance in urine is seen in phenylketonuria, in the galactosemia, and in the pancreas anemia. Very good, very good. So I'm so happy that all of you are able to give the correct answer. Great. Okay, let's move on to next question. Which of the following pairs of sugars are both non-reducing? We are again done this part also when you're talking about reducing and non-reducing. Which of the following pairs of sugars are both non-reducing? Your options are sucrose and lactose, sucrose and maltose, sucrose and trehalose, sucrose and isomaltose. What should be the correct answer? What should be the correct answer? We know that lactose is reducing, maltose is reducing, isomaltose is reducing, sucrose we know is non-reducing, and trehalose we had written was non-reducing. So your correct answer is C. Very good, very good. It is so nice to see that most of you are able to give the correct answer. The correct answer is sucrose and trehalose. Sucrose and trehalose are a pair of sugars which are both non-reducing. Sucrose and trehalose both are non-reducing. Okay, so now we move on to the next question. UDP glucose is used for all except, now again this is something we have not yet discussed, but I assume that most of you should be able to this to answer this question. Let's see how many of you are able to attempt this question and give us the correct answer. UDP glucose is used for all except. What is the correct answer? UDP glucose is used for all of the following except. Okay, please note the UDP glucose, the UDP glucose will, okay, I can see some answers coming in. UDP glucose not used for heparin, it is not used for galactose metabolism. What else? What else? UDP glucose can get converted to UDP glucuronic acid. Similarly, UDP galactose, we'll see later, is converted into UDP glucose, and later we'll see in glycogen metabolism that UDP glucose is required for glycogen synthesis. I am sure you know this, that UDP glucose is required for glycogen synthesis. So what is happening here? UDP glucose required for glycogen synthesis, UDP glucose required for galactose metabolism. The UDP glucuronic acid is required for the synthesis of glycose aminoglycans. It is required for synthesis of glycose aminoglycans, and remember heparin is also a glycose aminoglycan. Heparin is a glycosaminoglycan. It requires the uronic acid. We had said amino sugar and uronic acid are required for the glycosaminoglycan. One of them was heparin. So heparin also requires UDP glucuronic acid. So the correct answer in this case should be the sphingolipid synthesis. In the sphingolipid synthesis, the UDP glucose is generally not required. Generally, it is not required. Similar question again from the glycosaminoglycans and bit of applied aspect. Glucosamine used in the treatment of remember the glucosamine we had said is required for the synthesis of glycosaminoglycans. The glycosaminoglycan have a very important role in synovial fluid, replace the synovial fluid in arthritis. So I'm happy to see that a lot of you have been giving the correct answer. The answer being arthritis. Glucosamine used in the treatment of arthritis, it promotes the synthesis of the synovial fluid. It promotes the synthesis of the synovial fluid. So glucosamine is the correct answer. Glucosamine is used in the arthritis. Very good. It's very nice to see most of you have given the correct answer. Let's move on to the next question. It is an image based question. Following are the tests done for patients for proteins. Protein number one, sugars number two, and ketones number three. Before you give the answer, can you tell me which test is number one, which test is number two, and which test is number three? What are the tests displayed in the different images? The tests displayed in the different images. Anyone? What is one? What is two? What is three? Can you tell me the tests being displayed in number one, two, and three? Okay, so number one is the test for proteins. It is the biuret test. Number two is the test for reducing substance. Very good. Two is the Benedict's test. And three is the Rothera's test. The three is the Rothera's test. I'm sure you already know. Uh, I'm sure you already know what are the different tests used for. We already mentioned Biuret test is for proteins. The Benedict test is for sugars, and the Rothera's test is for the ketones. We already mentioned this. Okay. Now you have to tell me what will happen in the starvation. In a patient of starvation, what will you find in the urine? Will protein be there? No, protein will not be there. In starvation, no protein. Will sugar be there? No. Starvation, hypoglycemia, no sugar. Will ketone bodies be there? Yes, the ketone bodies can be there in patient of starvation. More and more of ketone bodies will be formed. It is a condition of ketosis which subsequently gives rise to the ketoacidosis. So the correct answer is only the ketone bodies in the urine, which is detected by the Rothera's test. It is detected by the Rothera's test. See, these image-based questions can come in your exams. What we'll do once we've completed our discussion of the carbohydrates, I'll show you some of the important image-based tests for the carbohydrates. Very good. Look at the next question. Most important rate limiting enzyme of glycolysis. The most important rate limiting enzyme of glycolysis: A. PFK1, B. Pyruvate kinase, C. Glucose 1,6 bisphosphatase, and D. Hexokinase. What should be the answer to this question? Most important rate limiting enzyme. See, I had mentioned previously also, this is the most frequently asked question. The most important rate limiting enzyme for any pathway. Most important rate limiting enzyme for any pathway. This is the most commonly asked question. And obviously, I'm sure you are able to give the answer. Very good. PFK1 is the correct answer in some guides and books. Hexokinase is given, which is not correct. Please note, PFK1 is the enzyme. Sometimes the question can be, all are rate limiting enzyme of glycolysis except. In that case, what are you going to mark? All are rate limiting enzyme of glycolysis except. In that case, going to mark. I've changed the question slightly, and now you have to give me the new answer. All are a rate limiting enzyme of glycolysis except. So what will be the answer in this case? Very good. So I'll again emphasize the point. In a pathway, there can be multiple rate limiting enzyme. In the question, you are generally asked about the most important rate limiting enzyme, but keep it in mind that there can be other rate limiting enzymes. So in glycolysis, all the enzymes which catalyze the irreversible reaction are rate limiting, and PFK1, pyruvate kinase, hexokinase, all of them are rate limiting enzyme. And in this case, the correct answer will definitely be glucose 1,6 bisphosphatase. Very great. So I hope the question answer session was useful to you for revising what we have covered in the last 3 days. If you have any doubts from the last 3 days session, please feel free to mention them. Else we are moving on to the metabolism of the glycogen from glycogen, and then the galactose and the fructose. I'll give you 2 minutes, and if you have any doubts, if you have any queries from what we have discussed in the last 3 days or from the questions that we have covered just now, please feel free to ask them. You can also ask from any extra information that you want. We'll give you 2 minutes. Quickly send across your questions, doubts, and any extra information that you need. If there are no queries or doubts, just give me a thumbs up so that I know we can go ahead. Okay, I'll presume that we don't have any questions, and I'll move on to our next topic, that is the glycogen. I'll move on to our next topic, that is the glycogen. Quick recap what we had talked about glycogen previously. We had mentioned that glycogen is the storage form of carbohydrate in animals, and it corresponds to starch in plants. It corresponds to starch in the plants. We had said it is a branched polymer of the D-glucose and it is known as the glucosan. We had said it occurs mainly in the it occurs mainly in liver and muscle with modest amounts in the brain. This is what we had mentioned. Modest amounts in brain. At the same time, we had said the muscle glycogen, similarly, the brain glycogen is providing energy to the muscle itself or the brain itself. It is meant for the local use. It is meant for the local use, whereas the liver glycogen is used to maintain the blood glucose. The liver glycogen used to maintain the blood glucose. The muscle glycogen is used for providing energy within the cell only. It is used for providing energy within the cell only, meaning the function of the glycogen in the different tissues is different. Please understand this fundamental point. The function of glycogen in the different tissues is different. In liver, it maintains the blood glucose. In muscle, it is only for local use. Now, before we go into the details of glycogen, there is a very important precursor molecule called glycogenin. Now you should know this because sometimes question can be asked on glycogenin also. Glycogenin is a protein. First point, what is glycogen? Glycogen is a protein. It is 37 kilodalton protein. It is glucosylated on a tyrosine residue. This question is asked, on which amino acid the glycogenin is glucosylated, and the answer is the tyrosine residue by the UDP glucose. The result the result is formation of a glycogenin primer. The result of glycogen primer, that is the precursor of glycogen. You have to start from somewhere, so you first make a small structure which resembles the glycogen. This is known as glycogen primer. Please note that this glycogen primer in the beginning is attached to the glycogenin. In the beginning it is attached to the glycogenin. Once the glycogen primer is formed, it can then be extended and elongated to give us the final glycogen. Now here another difference is there. In muscle, all glycogen is attached to glycogenin. Reason being, the concentration per unit mass in muscle of glycogen is comparatively less. So there is sufficient amount of protein to be attached to all the different glycogen molecules. But in liver, it's not so. In liver, the concentration of glycogen per unit mass is very high. The glycogen may or may not it may or may not be attached to glycogen. It may or may not be attached to glycogen. So this is one very important point to remember. Glycogen is the you can say scaffold. It is a support on which the glycogen is formed. In muscle, all glycogen is attached to glycogen, but in liver, it need not be so. They may or may not be attached. Okay, once we know this, let's quickly take a look at the glycogen synthesis. Let us take a quick look at the glycogen synthesis. Okay, so we'll quickly take a look at the glycogen synthesis and then go back to our details. So, glucose is first converted into the glucose 6 phosphate. Glucose is first converted into glucose 6 phosphate. We already know the enzyme is glucokinase. Please note, in this case the enzyme is glucokinase. It cannot be hexokinase. I'll tell you later why it cannot be hexokinase. It is the glucokinase. The glucokinase will convert the glucose to glucose 6 phosphate. For this process it requires the ATP, and magnesium is required as the cofactor. We already done this part we've already done. But here what happens, this glucose 6 phosphate is quickly isomerized into the glucose 1 phosphate. The name of the enzyme is phosphoglucomutase. So phosphate is shifted from position 6 to position 1 by an isomerase enzyme. It is shifted from position 6 to position 1 by an isomerase enzyme, and here it attaches to UTP. It attaches to UTP to form the UDP glucose. It forms the UDP glucose. The pyrophosphate is thrown out. The pyrophosphate is thrown out. Alright, so glucose 1 phosphate has now been converted into the UDP glucose. The UDP glucose has two possible fates. Either it combines with the glycogenin, it combines or it attaches to the glycogenin. In that case, it will form the glycogen primer. Or if the glycogen primer is already there, if the glycogen primer is already there, in that case it forms the alpha 1 4 glycosidic linkage. It forms the alpha 1 4 glycosidic linkage, and this reaction is catalyzed by the glycogen synthase. This reaction is catalyzed by the glycogen synthase. So glycogen synthase will attach the UDP glucose to the glycogen primer, and this linkage is via alpha 1 4 linkage. The UDP glucose attached to glycogenin by the glycogenin itself. The glycogenin is the support also, it is the enzyme also. But what happens in this reaction? The UDP is released. The UDP is released, and this UDP, under the action, or you can say under the help from ATP, gets converted back to UTP. It gets converted back to UTP. All right. Gradually, as the chain becomes longer, as the chain becomes longer, it is acted upon by another enzyme, the branching enzyme. It is acted upon by another enzyme, and that is the branching enzyme. What the branching enzyme does? The branching enzyme adds, not important, forget it. I'll show you later, don't worry, leave it at that. So what is happening? The alpha 1,6 linkage is formed by the branching enzyme. So what is happening? Our glycogen is getting mature. The glycogen becomes mature, and at this stage it has both alpha 1 4 glycosidic bond and alpha 1 6 glycosidic bond. The alpha 1 6 glycosidic bond has been added by the branching enzyme. The alpha 1 6 glycosidic bond has been added by the branching enzyme. In this way, we get the mature glycogen. The mature glycogen is obtained, which has both the alpha 1 4 and the alpha 1 6 glycosidic linkage. The conversion to glucose 1 phosphate is a single step reaction. Directly by getting the phosphate from PLP. PLP will provide the phosphate. Under the influence of enzyme, glycogen phosphorylase. Under the influence of the enzyme, glycogen phosphorylase. The last glucose is quickly cleaved. The last glucose is quickly cleaved, and it gives us the glucose 1 phosphate. This process is known as phosphorylasis. This process is known as phosphorylasis, right? So phosphorylasis will cleave the last glucose to give us the glucose 1 phosphate. However, when the chain becomes shorter, at that time the glycogen phosphorylase is unable to act. We need to remove the chain. The chain is removed by the action of first the glucan transferase, the enzyme glucan transferase, and subsequently by the debranching enzyme. Subsequently by the debranching enzyme. A very important point to note. A very important point to note. The two enzymes are the enzymatic function of the same protein. They both are the both enzymatic function are present within the same protein. So I've made the same box divided to two. The two proteins are present. The two enzymes are the catalytic domain of the same protein. But what is very important to remember here is the debranching enzyme does not cause phosphorylasis, it causes hydrolysis. Result, free glucose is formed. Free glucose is formed by the action of the debranching enzyme. By the action of the debranching enzyme, the free glucose is formed, not the glucose 1 phosphate, because the debranching enzyme causes hydrolysis. It does not cause phosphorylasis. We get the free glucose. What happens once you have the glucose 1 phosphate? By the action of same enzyme, first we get the glucose 6 phosphate, and as you remember, the glucose 6 phosphate can be converted into glucose by the action of the enzyme glucose 6 phosphatase, which is associated with the release of phosphate as the inorganic phosphate. The phosphate is released as the inorganic phosphate. Okay, so this is the complete cycle of the glycogen metabolism. How, when there is lot of glucose, the glucose can be converted to glycogen for storage, and when there is need, this glycogen can be mobilized. This glycogen can be mobilized to give us back the glucose. In case of muscle, remember, the glucose 6 phosphatase will be there in the liver and the kidney only, it is not there in the muscle. So what happens in the muscle, the glucose 6 phosphatase is formed and it is diverted towards glycolysis. We had seen the glucose 6 phosphate obtained from glycogen is diverted towards glycolysis in anaerobic metabolism in the muscle. There, the free glucose will not be formed because the enzyme glucose 6 phosphatase is not there. The glucose 6 phosphatase is there in the liver. So, free glucose from glycogen will come only in the liver, not in the muscle or in the brain. We will not get the free glucose. Alright, few points to note. How many ATP? How many ATP are consumed when you are trying to uh, add one glucose to glycogen? How many ATP are consumed when you are trying to add one glucose to the glycogen molecule? Your options, I'll give the options because you always need options. Your options are 1, 2, 3, and 4. How many ATP required to attach one glucose to the glycogen? How many ATP required for attaching one glucose to the glycogen? Your options are 1, 2, 3, and 4. Okay, 3 ATP, 2 ATP, what else? 3 ATP, 2 ATP, what else? Very good, very good. So I'll highlight the answer. 1 ATP is used here. You can see 1 ATP is used here. The second ATP is used here. As you can see, the second ATP is used here. So the correct answer is 2 ATP. 2 ATP required for adding 1 glucose to the glycogen. 2 ATP required total to add 1 glucose to the glycogen. Remember, the UTP is getting recycled. It is getting recycled. So it will not be counted in the energetics because we are not having a loss of UTP. UTP is getting recycled. So it has not actually been used. It has not actually been used. The correct answer will be 2 ATP required to incorporate 1 glucose into the glycogen. Okay. Another question which is asked, among all the enzymes, which enzyme is common to glycogen synthesis and the glycogen breakdown? So I'm giving you a second question. I'm giving you a second question. Among all the enzymes of glycogen metabolism, which enzyme is common to glycogen synthesis and the glycogen breakdown? This question has been asked quite a few times, and you should remember it is the phosphoglucomutase. It is the phosphoglucomutase which is common to both glycogen synthesis and the breakdown. All other enzymes in glycogenesis and glycogenolysis are unique. All other enzymes in glycogenesis and glycogenolysis are unique. Okay, now I'll tell you a very important aspect, which is the hormonal regulation of glycogen metabolism. The hormonal regulation of the glycogen metabolism. I'll tell you the glucan transferrin debranching with an exam. I'll show you the diagram. Don't worry. Let me just tell you the hormonal regulation, and then I'll show you the diagram for the branching, debranching, glucan transferase. All of them. Don't worry. So the central regulatory molecule in this case, the central regulatory molecule in this case is the cyclic AMP. Now the cyclic AMP activates the glycogen phosphorylase, means it stimulates the glycogenolysis. At the same time, it will inhibit the glycogen synthase. So that cyclic AMP has dual action. It stimulates the glycogen phosphorylase and it inhibits the glycogen synthase. Now we have two important hormones, insulin, glucagon, and epinephrine. Now we already know the action of glucagon and epinephrine on glycogen synthesis and glycogen breakdown. If you want to pinpoint the action of insulin and glucagon on glycogen synthesis and breakdown, what we know is insulin will promote the glycogen synthesis and it will inhibit the glycogen breakdown. The glucagon epinephrine will have the opposite effect. They will inhibit the glycogen synthesis and promote the glycogen breakdown from this we can quickly determine the action of insulin and glucagon on the cyclic AMP. How are they going to affect the cyclic AMP? The insulin has to inhibit the inhibitor, then only the activation can occur. So what will cause? It will cause inhibition of cyclic AMP, whereas glucagon will cause activation of cyclic AMP. So all of these questions are asked independently. What is the effect of insulin on glycogen phosphorylase? What is the effect of insulin on glycogen synthase? What is the effect of insulin on glycogen synthesis and glycogen breakdown? And what is the effect of insulin on cyclic AMP? Similarly, what is the effect of glucagon epinephrine on glycogen phosphorylase? What is the effect of glucagon epinephrine on glycogen synthase? What is the effect of glucagon epinephrine on glycogenolysis and glycogenesis? And lastly, what is the effect of glucagon epinephrine on the cyclic AMP? These are 10 questions which can be asked, and you should know the answers to each of the 10 questions. 10 questions which can be asked, and you should know the answer to each of the 10 questions. Okay, so this was the hormonal regulation, but the hormonal regulation can quickly be overcome by allosteric regulation, particularly in the case of the glycogen phosphorylase. It shows allosteric regulation. This allosteric regulation is able to overcome even the hormonal regulation. So let us look at the allosteric regulation on glycogen phosphorylase, and then I'll quickly do the recap. Don't worry, I'll do the recap, but let us first finish the allosteric regulation on the glycogen phosphorylase. Remember, there are different isoenzymes of the glycogen phosphorylase in the different tissues. In the muscle and in the liver, we have different isoenzymes of glycogen phosphorylase. Whatever we are going to mention now, it relates to the allosteric regulation of glycogen phosphorylase. Okay, so first point, in both liver and muscle, in both liver and muscle, ATP and glucose 6 phosphate, ATP and glucose 6 phosphate will inhibit the glycogen phosphorylase. In both liver and muscle, ATP and glucose 6 phosphate will inhibit the glycogen phosphorylase. In liver, but not in muscle. In liver, but not in muscle. Free glucose will inhibit free glucose will inhibit the glycogen phosphorylase in liver but not in muscle. Similarly, in muscle but not in liver. In muscle but not in liver. Calcium and AMP will activate the glycogen phosphorylase. In muscle but not in liver, the calcium and AMP will activate the glycogen phosphorylase. If you want to look at the justification of these changes, it is very straightforward. See, what was the function of glycogen in the liver? It was to produce the free glucose. So if already a lot of free glucose is there in the liver, no need to break down the glycogen. Similarly, in the muscle, what is the function of glycogen? To provide energy. If AMP is there, it indicates low energy status. So we must provide energy for continued contraction, glycogen breakdown will occur. And calcium denotes ongoing muscle contraction. If ongoing muscle contraction is there, we don't know how much energy is going to be required. So we cannot stop the production of energy. So glycogen breakdown must continue if there is high amount of calcium and if the AMP concentration is high. Okay, so this was about the allosteric regulation of glycogen phosphorylase, how it is similar in the different tissues and how it varies between liver and in the muscle. One very important point related to the allosteric regulation, sorry, the hormonal regulation, which you have to remember. The hormonal regulation that we had seen should be applicable to both liver and muscle, but there is a very important point to consider. In muscle there are no receptors for glucagon. This means it doesn't matter for how long you have been fasting. It doesn't matter how high the level of glucagon is there. The muscle glycogen cannot be mobilized by glucagon. How will you mobilize the muscle glycogen? By epinephrine or by exercising, by increasing the calcium. So glucagon cannot mobilize the liver glycogen. The liver glycogen has to be mobilized either by exercise or by the epinephrine. When there is a fight and flight response, when there is fight and flight response, at that time the muscle contraction has to occur very fast, and that energy will come from glycogen, which is regulated by the epinephrine and not by glucagon. So remember, this is a very important point. In muscle there are no receptors for glucagon. In muscle there are no receptors for glucagon, so glucagon cannot regulate the glycogen breakdown in the muscles. Okay, let's go back and show you a few things. I'll show you the reaction per se also, and I'll show you the uh, some important images that I want you to see. Let's take a look what happened. Somebody was asking the rate limiting enzyme. Rate limiting enzyme is described for glycogenolysis, and the answer is the glycogen phosphorylase. Glycogen phosphorylase is the rate limiting enzyme in glycogenolysis. We already said distinct isoenzymes of glycogen phosphorylase in liver, muscle, brain, and they will behave differently. Okay, this is the whole diagram that we have already drawn. Here you will be able to see some extra enzymes which I had not mentioned previously. These are not required, but if you want you can note them down. The enzyme converting the UTP to UDP glucose is the UDP glucose pyrophosphorylase. The enzyme which is converting the UTP to UDP glucose is the UDP glucose pyrophosphorylase. Okay, that is one. The second enzyme which recycles the UTP, is the nucleoside diphosphokinase. The nucleoside diphosphokinase will recycle the UDP. It is converted to UTP by the action of nucleoside diphosphokinase. I'll again repeat, these enzymes are generally not asked, but if you want, you can note them down. The enzymes are generally not asked, but if you want, you can note them down. So UDP glucose pyrophosphorylase and the nucleoside diphosphokinase. Okay, I'll quickly go through this to have a recap, and then I'll show you some diagrams. We had mentioned starting from glucose. Glucose first converted to glucose 6 phosphate by the action of glucokinase, not hexokinase, where ATP is consumed. And this glucose 6 phosphate by the action of phosphoglucomutase is converted into the glucose 1 phosphate. Glucose 1 phosphate is quickly joined to the UTP, resulting in formation of UDP glucose. UDP glucose attaches to glycogen to form the glycogen primer. If the primer is already there, in that case it attaches via 1,4 glycosidic linkage. The UDP is released, which can later be recycled. The UDP, sorry, 1,4 glycosidic linkage increases the length of glycogen, and after some time there will be branching. The branching is done by the branching enzyme, resulting in the mature glycogen, which has the 1,4 linkages for the long chain, and branching side will have the alpha 1,6 linkage. The cleavage of the terminal glucose on glycogen is carried out by the glycogen phosphorylase by using a phosphate. I told you this phosphate will come from pyridoxal phosphate, also known as the PLP. Commonly, it is written as the PLP. Paradoxal phosphate will donate the phosphate for the action of glycogen phosphorylase. ATP is not required here, and we get the glucose 1 phosphate. If glucan transferase and the debranching enzyme come into play, I already said we get the free glucose, because here hydrolysis occurs, not the phosphorylasis. The glucose 6 phosphate converted back to glucose 6 phosphate, and the glucose 6 phosphate to glucose by the glucose 6 phosphatase, by the action of the glucose 6 phosphatase. So here I'll show you the diagrams for how the branching occurs and how the debranching occurs. Once I show that, after that you can ask any questions that you have. Okay, let's see. So the glycogen synthase will add the glucose residues to the two chains, and the chains will keep becoming longer. Once the size of the chain is at least 11 residues long, the minimum size at which the branching can occur is 11 residues. So once the size of the chain is minimum 11 residues long, that is when the branching enzyme will come into play, and it shifts the last 6 residues. It shifts the last 6 residues. If you want, you can count 1, 2, 3, 4, 5, 6. 6 residues are shifted to an adjoining chain, and in this case the linkage which is formed is alpha 1,6 glycosidic linkage, which is denoted by a dash. Wherever you find the dash, it will show the alpha 1,6 linkage, will show the alpha 1,6 linkage. Okay. Second diagram that I want to show you is for the debranching enzyme, the action of the debranching enzyme. So the phosphorylase, when it starts acting, it will break the chains, it will keep making them smaller. But when we have a very small chain on the branch side, 4 residues only, at that time the phosphorylase cannot act any longer, and that is when the glucan transferase will come into play. It shifts the last 3 residues from the 4 residues to an adjoining chain. So what you're left with is only the branch site. We are left with only the branch site. The branch site is cleaved by the debranching enzyme. Remember, the reaction which is occurring here is hydrolysis, and you get the free glucose. This glucose is not phosphorylated. What you get is the free glucose. Okay, we already talked about the difference in the glycogen phosphorylase at different levels. What I want to mention very quickly is the glucokinase. Why we have the glucokinase and why not the hexokinase? Then why the glucokinase and why not the hexokinase? Let's quickly compare the two. The hexokinase versus glucokinase. Let us quickly compare the two. Okay, few points on which we must compare the two isoforms or the two isoenzymes are the Km, the Vmax, location, hormonal regulation, and allosteric regulation. And then the picture will become very clear. Okay, let me complete this and then I'll go back to the debranching enzyme. Let me complete this and then I'll go back to the debranching enzyme. Hexokinase has good affinity for the glucose. All right. So good affinity means low Km. We had seen the Km and affinity are inversely related. We had mentioned this. The Km and affinity are inversely related. So when I say good affinity, this means low Km. For glucokinase, the affinity is less. So Km is high. Now let's look at the Vmax and the allosteric regulation. Then the picture will become clear. Don't worry. The Vmax for hexokinase is also low. Reason is the feedback inhibition. Remember, as more and more glucose 6 phosphate will be formed by reaction of hexokinase, it will start inhibiting the enzyme itself. It will start inhibiting the enzyme itself. Therefore, the Vmax for the hexokinase is low, whereas there is no feedback inhibition in case of glucokinase, and therefore it is able to have a high Vmax. Hormonal regulation, no hormonal regulation for hexokinase, but increase in quantity of glucokinase occurs in response to increased glucose. In response to increased glucose, when the insulin will rise, it will result in more production of the glucokinase. Location: hexokinase is present everywhere. Glucokinase is present in the liver, and for muscle it is plus minus. It's not present in significant amounts. It is there but not in significant amounts. Now please try to understand, the hexokinase is the enzyme for basal metabolism. Means it should be able to act at the basal concentration, in the concentration range of 60 to 90 milligram percent. It should be able to act. Therefore it must have good or the high affinity. It must have good or high affinity, and that is why it has low Km. On the other hand, we said the glucokinase is used for making the glycogen. When do you want to make the glycogen? When the glucose is there in excess. How will we sense whether the glucose is there in excess or not? By having a low affinity. We will get activated only when the glucose level is high. So glucokinase will be able to act only when the glucose concentration is high. This ensures the glycogen synthesis occurs only when the glucose concentration is high. Alright, the hexokinase cannot act at high concentrations because of the feedback inhibition. when we are going to make the glycogen, at that time glucose concentration must be high. If the glucose concentration is high, we'll have more of glucose 6 phosphate and there will be feedback inhibition, and therefore sufficient glucose 6 phosphate which can derive the glycogen synthesis is not formed by the action of hexokinase. We require the glucokinase for that particular action. I hope this clears the point why glucokinase, why not hexokinase in the glycogen synthesis. I'll quickly repeat another point about the D branching enzyme. Let's go back and see the point for the D branching enzyme. All right. So two things to note. Number one, when the size of the branch chain is only 4 residues, at that time the glycogen phosphorylase cannot work. The glycogen phosphorylase cannot work when the size of the side chain is very low. It is up to 4 residues. At that time, the phosphorylase will not be able to work. So what happens? The glucan transferase comes into play and it removes the last 3 residues. It removes the last 3 from 4. So as you can see, the last 3 residues have been shifted from here to the chain, and now the branch site will get exposed. The branch site has gotten exposed and we cleave the branch. We are cleaving the branch, so we call the enzyme as the debranching enzyme. You are removing the branch, so it is called the debranching enzyme, but this debranching is done by hydrolysis, so we get the free glucose. Remember, this dashed line indicates alpha 1,6 linkage or the branch site, alpha 1,6 linkage or the branch site. Any questions so far? Before you go to the topic of the glycogen storage disorders, any questions so far? Okay, so now there are the rate limiting enzyme for glycogenesis is not defined. Rate limiting enzyme for glycogen synthesis is not defined. Reason being, we only have two enzymes in this whole picture. Only two enzymes. The I'm sorry, the glycogen synthase and the branching enzyme. So if you talk about glycogen synthesis, effectively there is only one enzyme, that is the glycogen synthase, because the branching enzyme will act when the glycogen chain has been elongated. So for glycogen synthesis, we don't have a rate limiting enzyme. For glycogenesis, we don't have a rate limiting enzyme because the critical enzyme is only one. There is only one critical enzyme, glycogen synthase. So we don't describe the rate limiting enzyme for glycogen synthesis. Okay. Okay. Now, various may be deficient in different individuals. Their concentration may not be enough to maintain the glycogen metabolism, in which condition there can be defect in glycogen storage. Either defective glycogen will be formed or the glycogen which is being formed not be mobilized, resulting in increase in glycogen stores or increase in defective glycogen being stored in the body. So I'll quickly highlight those enzymes. in this pathway, and then we will tabulate them. No need to write them here. The first is the deficiency of the enzyme glucose 6 phosphatase. The first is the deficiency of the enzyme glucose 6 phosphatase. This is known as the von Gerke's disease. This is known as the von Gerke's disease. I'll talk about the number 2 disease later. Don't worry about that. Then in some individuals there is a defect of the D branching enzyme. This is the Cory's disease. We'll write that, don't worry. This is the Cory's disease, the glycogen storage disorder type 3, where the D branching enzyme is missing. In some individuals, the branching enzyme is missing. This is also known as the Anderson's disease. This is also known as the Anderson's disease. Now, in some individuals, the muscle phosphorylase, and some individuals the liver phosphorylase can be deficient. When the muscle phosphorylase is deficient, that's number 5. When the liver phosphorylase is deficient, that's number 6. Glycogen storage disorder number 5, number 6. Deficiency was number 1. Defect in the debranching enzyme was number 3. Defect in the branching enzyme was number 4. Alright, we also have a number 7, which I'll show you shortly. Okay, so let's give the names to these numbers. Nowadays we use numbers, but they also have old traditional names. Each of them have old traditional names. So we'll go to the glycogen storage disorder and give them the names. Like I said, the GSD type 1, previously known as the von Gerke's disease, previously known as the von Gerke's disease. And in this case you already seen which enzyme is deficient. We already seen the enzyme which is deficient. The number 2 is known as the Pompey's disease. Now this disease is different from all the different glycogen storage disorders. A reason being, here the part of glycogen metabolism which is defective is not the one that we discussed. Remember, when I was mentioning glycogen, I told you some amount of glycogen synthesis occurs in all the tissues, but not significantly enough that we can store it. Now, this glycogen synthesis occurs within the lysosomes. So, glycogen synthesized lysosome, metabolized, synthesized, metabolized. Sometimes that pathway can be defective, and that is seen in Pompey's disease. Lysosomal enzyme is defective, which cleaves the alpha 1,4 and alpha 1,6 bond. It is known as alpha 1,4,1,6 glucosidase. The common name is the acid maltase. Lysosomal acid maltase is defective in Pompey's disease. 3, third alphabet, Cory's. You can remember in this way. The name of GSD3 starts with the third alphabet, and that is Cory's. And the corresponding enzyme defect is the letter after C. C is followed by D. So the defective enzyme is the D branching enzyme. C followed by D. So defective enzyme is the D branching enzyme. Just to complete the record, I'll write the name of this enzyme, although it was not required. Similarly, we come to 4. The name of the disease is Anderson's. What is the letter after A? After A, we get the B. So defective enzyme is the branching enzyme. C followed by D and A followed by B. C followed by D and A followed by B. The name of the fifth disorder is McArdle's disease. And here the defective enzyme also starts with M. In McArdle, the defective enzyme is also starting with M. We call it the myophosphorylase. We written behind the phosphorylase in the muscle. Phosphorylase in the muscle, that is the myophosphorylase, is defective. Similarly, in 6, the name of the disease is Her's disease. And here also the name of the defective enzyme starts with H. It is the hepatic phosphorylase. It is the hepatic phosphorylase. H for H, M for M. A followed by B, C followed by D. The last name that is mentioned in this list is actually not related to glycogen metabolism, but it results in glycogen accumulation. This is known as Tharui's disease. And here the defective enzyme is PFK1 only in 2 locations, in RBC and muscles. Obviously in RBC there is no glycogen, but in muscle the glycogen stores will not be used up properly, so there the glycogen accumulation will occur. So these are the 7 important glycogen storage disorders which you must know about, and if you can, you should remember the name of the enzymes. I hope the names are quite easy to remember. The first name all of you know, von Garke's defective enzyme is the glucose 6 phosphatase. 2 generally not touched. In 3, C followed by D. In 4, A followed by B. Scories, D branching, Anderson's branching. In 5, M for M, McArdle's disease, defective enzyme, myophosphorylase. In 6, H for H, Her's disease, hepatic phosphorylase. 7th is different from everyone. In Tharui's, the PFK1 is defective, meaning what you'll find is the defective glycolysis. Whether it is aerobic or anaerobic, we'll find the defective glycolysis in RBC and in the muscles in these patients. Now, 2 things. Number 1, when are you going to suspect the glycogen storage disorders? What is the condition? What are the findings which will make you start suspecting the glycogen storage disorders? So let's take a quick look. When are you going to suspect the glycogen storage disorder? 3 things will help you to quickly identify the glycogen storage disorder. Number 1, when you examine the patient, you find hypoglycemia. And number 2, you find biochemical signs and symptoms which indicate that hypoglycemia is chronic. Biochemical signs which indicate that the hypoglycemia is chronic. What are those signs? Elevated free fatty acid, elevated cholesterol, elevated VLDL, elevated ketone bodies. All these will indicate that the hypoglycemia is chronic in nature. See, when the hypoglycemia is there, the fat stores are mobilized, so therefore you should have the fatty acid in the larger amount. This fatty acid will go to the liver where it is converted to the cholesterol, so you should have more of cholesterol. In addition, it is packaged back into the triglycerol and released in the form of VLDL, so you can find increased VLDL. And lastly, this fatty acid which is being mobilized will also be converted into the ketone bodies. So what we'll find is the increased amount of ketone bodies also. All of these are biochemical signs of chronic hypoglycemia. Biochemical signs of chronic hypoglycemia. And third one, at least one organomegaly must be described. At least one, whether it is abnormal facies, whether it is enlargement of the liver, the kidney, the muscle, at least one organ enlargement must be there. If you find these three things, then you start suspecting, the glycogen storage disorder. But how to distinguish between the different glycogen storage disorder? How to distinguish between the different glycogen storage disorders? So let's take a quick look how to distinguish between the glycogen storage disorder. Okay, so what I'll do, I'll mention the important features for each of them. I'll write them by number rather than going with the name. I'll write them simply by number. Let's start with GSD type 1. GSD type 1, is the von Gerkes disease. GSD type 1, is the von Gerkes disease. So in addition to the three things that we mentioned, which tells us that we'll probably looking at the glycogen storage disorder, what you'll find here is severe hypoglycemia. This severe hypoglycemia can cause fainting episode if the individual does physical activity in such a state. So what you'll find is fainting episodes with exercise during fasting. Later on, we'll also find one important biochemical finding here, that is hyperuricemia. We'll discuss this when we are doing the purine metabolism. The hyperuricemia is seen in glycogen storage disorder, and because the glucose 6 phosphatase was defective, it is present only in the liver but not in the muscle. What you find is hepatomegaly only. Hepatomegaly will be there, but muscle enlargement will not be there. Hepatomegaly will be there, but muscle enlargement will not be there. The most characteristic finding is the fainting episode. The most characteristic finding is the fainting episode. Okay, so GSD1, history of fainting episode, and the severe hypoglycemia. In addition, the features of chronic hypoglycemia that we mentioned, they may be described in the question. For GSD2, two things: decreased lifespan. The question may give history of early death in the patient itself, or it may give the history of early death in a sibling. In a previous sibling, it may describe that a sibling expired previously by the age of 3 years. In addition, you'll find multi-organ involvement. Even the bones of the face are involved, resulting in abnormal facies. Bones of the face are involved, resulting in abnormal facies. In GSD3, remember the defective enzyme was the D branching enzyme. So what happens when we take a meal? The glycogen is formed, the branch sites are formed. But when we enter the fasting stage, the chains will become short. But when it comes to removing the branch site, the D branching enzyme is not working. So the branches will remain intact. And when you eat again, more and more branches will be formed. So what we find? Highly branched glycogen. When you take the biopsy, you'll find highly branched glycogen. This highly branched glycogen resembles a compound called limit dextrin. This highly branched glycogen resembles limit dextrin, and therefore the condition is known as limit dextrinosis. The condition is known as limit dextrinosis. In addition, you'll find both muscle and liver enlargement. All right, both muscle and liver enlargement. In GSD4 also, we will have the muscle and liver enlargement. Both muscle and liver are enlarged, but here we find poorly branched glycogen. We find the poorly branched glycogen. Now, this poorly branched glycogen looks like the amylopectin of the starch. We had said that amylopectin of the starch is also branched, but it is poorly branched. So, it resembles amylopectin, and therefore the condition is known as amylopectinosis. The condition is known as amylopectinosis. So poorly branched glycogen resembles amylopectin, and the condition is known as amylopectinosis. Now we come to the GST type 5, the McArdles. Before we discuss the sign and symptom of McArdle, we should understand the physiology of muscle contraction. There is an overlap with physiology, so I'll quickly do the recap which is relevant here, and then we'll come back to the presentation of GSD5. What we need to know is the source of energy. Source of energy for muscle contraction. Source of energy for muscle contraction in different stages. In different stages of contraction, what are the source of energy? So we describe the stages as the first 3 to 5 seconds. All right. In this stage, the source of energy is stored ATP. In this stage, the source of energy is the stored ATP. Subsequently, the next 20 to 30 seconds, in the next 20 to 30 seconds, the stored phosphocreatine, the stored phosphocreatine is used to regenerate the ATP. The stored phosphocreatine is used to regenerate the ATP, and this phosphocreatine is known as phosphagen or the high energy phosphate. It is known as the phosphagen or the high energy phosphate. Now, when you start doing the exercise, the neural impulse goes to the ANS that contraction is going on and we need more and more blood supply. So, after some time, the vasodilatation will occur, but this vasodilatation takes some time to appear. So, after the stored phosphocreatine has also been used, but vasodilatation has not occurred. So, we are talking about the phase before vasodilatation, but after the stored phosphocreatine has been consumed. After the stored phosphocreatine has been consumed. So in this stage, the glycogen will be used as the fuel for breakdown and providing the energy, because the sufficient extra blood is not there to meet the extra energy requirement. Remember, in this case, the glucose which will be released will undergo anaerobic glycolysis. The glycogen will be broken down by the action of the phosphorylase. We are talking about muscle contraction, so it will be broken down by the myophosphorylase. Once the vasodilatation occurs, once the vasodilatation occurs, then the energy will be obtained from blood glucose. And in this case, what do you see? We see the aerobic glycolysis, because when the blood supply increases, the available oxygen will also increase. So we find aerobic glycolysis, right? Now let's talk about the condition type 5. Type 5, where the myophosphorylase is not there, and this person starts to do the exercise. This person starts to do the exercise. You can take the example of yourself. Suppose in your city the marathon is going to be held 3 months from now. So you tell one of your friends who participates in the marathon every year. He participates in the marathon every year. So you tell him, okay, dear friend, this time I am also going to participate in the marathon. So how do I go about it? So your friend tells you that, okay, because marathon is a long race, you cannot just get up in the morning and run 16, 18, 20 or the full 41 kilometers. You'll have to practice regularly and gradually build up your stamina. Only then you can complete the marathon. So obviously, so what you do, you have to start practicing. So today you've decided that you're going to run the marathon after 3 months. So tomorrow you start practicing. So tomorrow when you get up in the morning, you go with your friend and you start running. When you start running, in the first phase for 3 to 5 seconds, stored ATP is there. You are able to do the exercise. You can run. When you enter into stage 2, when the stored phosphocreatine is going to be used, you have enough stored phosphocreatine and you are able to keep running for 20 to 30 seconds. But when you enter stage 3, remember, in myocardials, the myophosphorylase is not there. So the glycogenolysis in the muscle will not occur. If it does not occur, the glycolysis cannot occur, and suddenly there will be no energy for muscle contraction, and you'll sit down. Your legs will give way, and you'll sit down. You'll say, "Okay, enough, done for today. We don't want to run more." Why? Because you don't feel any energy in your legs. All the energy has been exhausted. No new energy is coming, and then your friend will start laughing. He'll tell you, "Yeah, you can complete the marathon only in two ways. One, you run the whole 41 kilometers in 30 seconds because that is how long you can run. Is it possible? No. Then the second option is you have to practice. If you don't practice, you will not be able to complete the marathon." So you said, "Yeah, no, I cannot run. My legs are not working." So your friend will tell you, then you cannot run the marathon. So you think, okay, doesn't matter. Let's try to run. Now see what is happening here. When you start the muscle contraction, the signal for vasodilatation has gone. It appears after 2 to 5 minutes, but if you stop the exercise, it doesn't mean the vasodilatation will not occur. The stimulus is already gone. The vasodilatation will occur and then gradually resolve once you stop the exercise, but it will take time. So what happens when you get up after 2 3 minutes and you decide, okay, nahi, I have to do the exercise, because if I don't do, then I cannot run the marathon. And you enter the stage 4, you try to do the exercise, because vasodilatation has occurred, blood glucose becomes available, you are able to do the exercise. Inability to do the exercise in stage 3 after 30 seconds. This is known as exercise intolerance. So, one of the characteristic features of the myocardials is exercise intolerance. But the beauty is, after some time, this person can start doing the physical activity again without any limitation, and we call this the second wind phenomena. So the characteristic finding in a type 5 is the exercise intolerance followed by the second wind phenomena. This is the characteristic finding for a type 5. Remember, the defect is only in the muscle. Defect is only in the muscle. So only muscle enlargement occurs. Muscle enlargement, and I told you exercise intolerance with second wind phenomena. Exercise intolerance with second wind phenomena. I'll repeat again. See, in stage 1, the energy comes from stored ATP. In stage 2, the energy comes from stored phosphocreatine. In stage 3, the energy comes from glycogenolysis, for which we require the myophosphorylase, which will then undergo the anaerobic glycolysis. In stage 4, the energy comes from blood glucose, which is coming due to vasodilatation, and the energy will come from aerobic glycolysis. The energy will come from aerobic glycolysis. In patients of type 5, the glycogenolysis cannot occur because the myophosphorylase is not there. Stage 3 will not work. This will cause exercise intolerance. But after the vasodilatation, the individual can again do the work. This is known as the second wind phenomena, which is characteristic of the type 5, the McArdle's exercise intolerance with second wind phenomena. Okay, let's go to GSD type 6. GSD type 6 is the Hertz disease. Remember, Hertz disease. It is limited to the liver because we had said the hepatic myophosphorylase is defective. So here we find only hepatic enlargement. Obviously, hypoglycemia will be there, but it is not as severe as type 1. So no additional findings except the common findings of GSD. Now we come to the last one, which is the GSD type 7. Remember, in GSD type 7, we had mentioned the defective enzyme is the PFK1. So let's go here. Now, this was a type 5, and now we are talking about type 7, where the defective enzyme is the PFK1. So there's another person who's affected by the Tarui disease, the type 7, and he also wants to participate in the marathon. So when he starts running, he's able to run for the first 3 to 5 seconds. He's able to run for the next 20 to 30 seconds. But when he goes to stage 3, the glycogenolysis is okay. The glycolysis part is defective. So again, what we'll find here, the exercise intolerance. You find the exercise intolerance. All right, but when you go to stage 4, blood glucose has increased. How will it be metabolized? By glycolysis. Glycolysis cannot occur. PFK1 is not there. So here we don't have the second wind phenomena. So how do you distinguish between type 5 and type 7? Both of them will present with exercise intolerance, but in type 5 you'll find the second wind phenomena. In type 7 there will be no second wind phenomena, because here also the defective enzyme is defective only in the muscle. So if you look at enlargement, you find that only the muscle enlargement in type 5. Just like in type 5, only the muscle enlargement will be there. So this is how you can distinguish between the different GSDs. I'll quickly go through that. First, you have to determine that, okay, you're looking at the glycogen storage disorder. Three features which will help you to determine that you're looking at glycogen storage disorder. First, the finding of hypoglycemia. This is common to all the glycogen storage disorders. In addition, biochemical signs of chronic hypoglycemia. They are increased free fatty acid, increased cholesterol in the blood, increased VLDL in the blood, and increased ketone bodies. And lastly, any type of organomegaly, whether it is liver, whether it is a muscle, whether it is a kidney, all of them, uh, one of them will be elevated. In GSD type 1, in GSD type 1, we find the severe hypoglycemia, fainting episode with exercise during fasting, hyperuricemia, hepatomegaly is present. Along with this, we can also find kidney enlargement. We can also find the kidney enlargement because the glucose 6 phosphatase, the defective enzyme, is normally required in the kidney for the gluconeogenesis. So there also some intermediates will be formed, resulting in the kidney enlargement. In GSD type 2, the most important point that we need in the question is the decreased lifespan. In addition, multi-organ involvement will be there. There can be hepatomegaly, there can be abnormal fishes, there can be splenomegaly. So all that can be there. In GSD type 3, we find the highly branched glycogen. The condition is therefore also known as limit dextrinosis, and both muscle and liver are enlarged, just like in type 4. Type 3 is the Cory's, type 4 is the Anderson's. C followed by D, the D branching enzyme is defective, and Anderson's A followed by B, the branching enzyme is defective. In GSD4, we find the poorly branched glycogen, and that is why the condition is known as amylopectinosis. The condition is known as amylopectinosis. In GSD5, we find the muscle enlargement and exercise intolerance with second wind phenomena. In type 6, we find the hepatic enlargement with the characteristic findings of glycogen storage disorder. And in type 7, we find the second wind phenomena. Sorry, exercise intolerance without the second wind phenomena. So this is how you are able to distinguish between the different glycogen storage disorders type 1, 2, 3, 4, 5, 6, and 7. If you have any doubts, if you want me to repeat any part, please feel free. I'm giving you 2 minutes. Just take a quick look and let me know any part you want me to repeat. No questions? Any questions? If you have no questions, give me a thumbs up. Give me a thumbs up if there are no questions so that we can move on to the HMP shunt. If there are no questions, give me a thumbs up. We'll move on to the HMP shunt. No questions? Okay, great, great, great. Let's move on to the HMP shunt. Okay. Second wind phenomena repeat. Second wind phenomena repeat. Very good. Okay. Okay, so I'll quickly mention the second wind phenomena again. This is the second wind phenomena. So, in simple terms, what it means, initially you want to do some activity, but you're not able to do, but after some time you again feel energized and you are able to do the activity again. This is called the second wind phenomena. So, what is happening? I have described the contraction in different stages. You can see I've described the contraction in different stages. Okay. Now, in patient of type 5, which we call the McArdles, in McArdles, the defective enzyme, is the muscle phosphorylase, which will break down the glycogen in the muscle. That is the muscle phosphorylase. All right. So muscle phosphorylase is not working. So what will happen here? The glycogenolysis will not occur. All right. So glycogenolysis is not working. So stage 3 contraction in stage 3 cannot occur. There is no source of energy, and this will present itself as exercise intolerance. Person cannot do the physical activity after 20 to 30 seconds when the stored phosphocreatine is exhausted. But when you start doing the physical activity, the stimulus goes to the ANS for vasodilatation. All right. And even if you stopped doing exercise after 30 seconds, that stimulus is already gone. Okay. And the response will appear. And what is the response? The response is vasodilatation. Now, when the vasodilatation will occur, the blood supply will increase. When the blood supply increases, we'll get more blood, and along with that extra blood, we'll also get the extra glucose. The blood supply in the resting stage is enough to meet the basal requirement. It cannot sustain the enhanced contraction. But when there is vasodilatation, in that case, the blood supply increases, and the glucose present in this extra blood can now support the contraction. And it has to be metabolized by aerobic glycolysis. It will provide the energy, and now new muscle contraction can occur. This is known as second wind phenomena. Ability to do exercise after an intervening phase where you can't do the exercise. In stage 3, we said no exercise. But after you rest for some time, you are able to do the exercise. This is known as the second wind phenomena. You are able to do exercise after break of some time. This is known as the second wind phenomena. I hope it clears it up. What do you mean by second wind phenomena? I hope this is now clear. The glycogen storage disorders are very frequently asked in your NEET and the AIMS exam. So you must be very clear about the glycogen storage disorders. If the doubts still persist about any of the points, please don't hesitate to ask for clarification. Any more questions? In normal individuals, the second wind phenomena is not required. Please understand, in normal individuals, because the glycogen stores are there, glycogenolysis will occur normally. Therefore, the second wind phenomena is not required. It will be required only when the glycogen breakdown is defective in the muscles. So, in normal individuals, it is not required. Okay. In GSD type 2, we said this is the defect in the lysosome. Now, what happens here? Lysosomal glycogen synthesis and breakdown. Where does this occur? Where does this occur? It occurs in all tissues. The lysosomal glycogen synthesis and breakdown occurs in all the tissues. But now what is happening in GSD2? The lysosomal defect is there. and breakdown will not occur. The breakdown will not occur. So what will happen? This lysosomal glycogen synthesis will keep occurring, and increased glycogen stores in lysosome of all tissues. Not only the lysosomal accumulation, the glycogen breakdown provides energy which is required for the degradation of the proteins, and if the energy is not available, degradation of the proteins will start getting affected, and the life of the cells will decrease. So what happens? Accumulation and increased cell death in all tissues. So that's why I said multi-organ involvement. Here we had used the term multi-organ involvement, because all the tissues will start getting affected. All the tissues will start getting affected. So we had said the multi-organ involvement. Normally, the amount of glycogen being synthesized in the lysosome is very small, insignificant, and in normal individuals, we don't even need to mention it. But it becomes significant when the breakdown is defective in the glycogen storage disorder type 2. Otherwise, it is insignificant and need not even require a mention. In type 7, there is defect in PFK1 in RBC and the muscle. Now, PFK1 is the enzyme of glycolysis. When there is the breakdown of the glycogen in, let's say, during exercise, it has to go through glycolysis only. If glycolysis is not able to utilize, if it is not able to utilize the glucose 6 phosphate will start accumulating. And remember, the glucose 6 phosphate is an inhibitor of glycogenolysis. So, what will happen here? PFK1 is not working. Result, glucose 6 phosphate will increase, and this glucose 6 phosphate in turn is going to inhibit the glycogenolysis. You already said the glucose 6 phosphate and ATP will inhibit the glycogenolysis in all tissues. Allosteric regulation overcomes the hormonal regulation. Even if you are doing the exercise, the glucagon, epinephrine, calcium, they are coming into play, but the glucose 6 phosphate will inhibit the glycogenolysis. The glycogen will stay as it is in the tissue, it will not undergo breakdown. It will not undergo the breakdown. Okay. Okay. What else do we have? What else do we have? Any specific name for facies? The facies are described as they are described as coarse facies. This is common for lysosomal storage disorders. The term used is coarse facies, which is used for the lysosomal storage disorders. Common term, the coarse facies used for the lysosomal storage disorders. So I presume it is now clear to everyone, and let's go on to the HMP shunt. Okay, so we have completed the glycogen metabolism. We'll quickly go through the HMP shunt. HMP stands for the hexose monophosphate. It stands for the hexose monophosphate shunt. Hexose monophosphate shunt, also known as the pentose phosphate pathway, also known as the pentose phosphate pathway. Right, the HMP shunt, also known as the pentose phosphate pathway. Please note that we call it a shunt. It is a minor pathway. Only when glycolysis is saturated, this shunt will come into play, and we describe it under two phases. We describe this shunt under two phases: the oxidative phase and the non-oxidative phase. The oxidative phase and the non-oxidative phase. As you can see, oxidative phase is non-reversible, but it gives us the very important NADPH. The non-oxidative phase is reversible, and it gives us the pentose sugar, that is the ribose 5 phosphate. We'll see later in purine pyrimidine synthesis, the ribose 5 phosphate is required for the purine and pyrimidine synthesis both. The ribose 5 phosphate is required for the synthesis of both purine and pyrimidine. Okay, so let's go to the checklist for HMP shunt. The checklist for HMP shunt, but before we go to checklist, 2 points. What is the requirement of NADPH? NADPH has dual function. It requires reducing potential in synthetic process. So wherever anabolic activity is going on, you're synthesizing the hormones, you're synthesizing the fatty acid, you're synthesizing the cholesterol. Wherever anabolic activity is going on, we require the NADPH. All right. Second, NADPH is the most important antioxidant. So where oxidative stress is higher, there NADPH has very important role. So these are two primary functions of NADPH, and HMP shunt is the primary source of NADPH. Okay, so let's do the checklist. We'll come back to the reactions later. You can see these are some of the functions of the NADPH: fatty acid biosynthesis, the steroid synthesis, the cholesterol synthesis, the detoxification, the drug metabolism, to reduce glutathione, in WBC functioning, the oxidative burst, and in RBC membrane stabilization. All right. So synthetic activity as well as the antioxidant action. So in our checklist, the point number one is location or the organ where this is working. So the location is the same as where these synthetic processes are operating. So we'll quickly list some organs where NADPH is required in larger amounts and HMP shunt is upregulated. HMP shunt is upregulated in the testis, where we need the sex steroids. In thyroid, the thyroxine hormone is formed. In adrenal cortex, the different hormones are being formed. In adipose tissue, for lipogenesis. In liver, for widespread synthesis. In lactating breast, again for lipogenesis. In erythrocyte and in the eye and lens, their main role is the antioxidant function. Whereas in others, the main role is the biosynthetic function. The main role is the biosynthetic function. Okay, no worries. We have enough time. We can discuss in detail. Sometimes we will need to do the repetitions, but it shouldn't be a worry for others. You can use that for revision. Okay. Chalo. So these are the organs where HMP shunt is upregulated. Remember, it will occur in all the tissues. Some amount of HMP shunt is occurring in all the tissues. In these tissues, it is upregulated. It doesn't mean HMP shunt does not occur in other tissues. It doesn't mean the HMP shunt does not occur in other tissues. It does occur in all the tissues. So this is point one, location, and point two, the compartment, that is cytosol. The point two compartment is the cytosol. Here only I will mention the substrates, which is point number three. The substrate are three molecule of glucose 6 phosphate. We start off with three molecule of glucose 6 phosphate, and then let's see, sorry, for number 4, the products. The products are 2.5 molecule of glucose 6 phosphate. The remaining half glucose 6 phosphate is released as carbon dioxide, and simultaneously 6 NADPH are generated. 6 NADPH are generated. Alright, so the product is 2.5 molecule of glucose 6 phosphate plus 3 carbon dioxide plus 6 NADPH plus H plus 6 NADPH plus H plus. Okay, so this is about the substrate and the end product. Substrate and the end product, see, mostly it is being recycled in the reversible phase. The glucose is again regenerated. It is the intermediates which are important, and along with that the generation of NADPH, which is important. Okay, number 5 and 6, rate limiting enzyme and target for regulation. Both are glucose 6 phosphate dehydrogenase. The rate limiting enzyme and target for regulation, both of them are glucose 6 phosphate dehydrogenase. I'm showing you this reaction, these two reactions, because this is where the NADPH is being formed. These are the two reactions where NADPH is being formed. Therefore, you must know the reactions. First, the conversion of glucose 6 phosphate to 6 phosphogluconate. The enzyme is glucose 6 phosphate dehydrogenase, and 6 phosphogluconate then further oxidize ribulose 5 phosphate. The enzyme is 6 phosphogluconate dehydrogenase. These are the two enzymes which generate the NADPH. This question is asked. NADPH in glycolysis in HMP shunt is generated by which of the following enzymes? So remember, NADPH generated by glucose 6 phosphate dehydrogenase and 6 phosphogluconate dehydrogenase. One very important point. One very important point. ATP is not synthesized. In addition, ATP is not consumed. Both hold true. ATP is neither synthesized nor it is consumed, meaning this process is ATP neutral. Neither it is synthesized nor it is consumed. So the process is ATP neutral. Please remember, the process is ATP neutral. Now, two additional very important points. In some individuals, the enzyme glucose 6 phosphate dehydrogenase is deficient, the G6PD deficiency. In these patients, the antioxidant activity will be reduced because If reaction 1 is not occurring, reaction 2 will also not occur. Therefore, the NADPH production will drop drastically. Antioxidant function is hampered. As a result of this, when there is oxidative stress, we'll find hemolysis. When there is oxidative stress, we will find hemolysis. So G6PD deficiency, also known as favism, results in hemolysis during oxidative stress. This is the most common enzyme deficiency disorder in humans. This has been described as the most common enzyme deficiency disorder in the humans. However, it is not very severe. It will present only when the individual is exposed to severe oxidative stress. Otherwise, it will not be seen. All right. Okay. Second thing is, this is the phase one. Remember, we had described the phase one oxidative and the irreversible phase, and the phase two, the reversible non-oxidative phase. Here you have to make note of one very important enzyme, and that is the trans-ketolase. We'll make use of this enzyme later. The trans-ketolase requires thymine pyrophosphate as coenzyme. Vitamin B1 is required as coenzyme, and therefore it is used to determine the deficiency of vitamin B1. The activity of this enzyme is used to determine the deficiency of vitamin B1. All right, so you have to make note of transketolase. Transketolase belongs to which pathway? It belongs to the HMP shunt. Which part? The non-oxidative reversible phase. It belongs to the non-oxidative reversible phase. So quick recap for the HMP shunt. In HMP shunt, we said the tissues where HMP shunt is upregulated. They are the testis, thyroid, renal cortex, adipose tissue, liver, lactatic breast, erythrocyte, and lens. The compartment where the synthesis occurs is the cytosol. The starting material, we said 3 molecule of glucose 6 phosphate. End product, we said 2.5 molecule of glucose 6 phosphate with 3 CO2 and 6 NADPH. The rate limiting enzyme and target for regulation are the glucose 6-phosphate dehydrogenase, and the process is ATP neutral. ATP is neither synthesized nor it is consumed. Lastly, we mentioned that G6PD deficiency is one of the most common enzyme deficiency disorders, also known as favism, and transketolase, an enzyme of the HMP shunt, is used to determine the deficiency of vitamin B1. These are the highlights of HMP shunt, which you must remember. If you remember this, reactions are not required. You'll be able to answer all the questions from this list. Okay. Now here we are needing to answer four types of question. We'll come across four types of question. I'll first mention those questions, and then we'll see how we can quickly answer those questions. Any questions from HMP shunt so far? Any questions from HMP shunt and the glycogen metabolism? I'll quickly talk about insulin and then go on to galactose and the fructose metabolism. Any questions so far? If there are no questions, then I'll quickly talk about the questions related to insulin. What are the different ways in which the question is asked on insulin? No questions? No questions? Okay, great. The questions which can be asked are, they can be straightforward question. For example, action of insulin, meaning which pathways are upregulated by insulin, which pathways are downregulated by insulin. We'll remember this by a table. We'll remember this by a table. Second question, we'll talk about fed and the fasting state. No, the function of NADPH are not important, but you should have a brief idea that where the NADPH are required. Broadly, if you remember, biosynthetic process and antioxidant activity, that is enough. The biosynthetic activity and antioxidant activity, if you remember these two terms, that is enough. Uh, Role in antioxidant activity we will discuss when we are doing the glutathione, but currently it is not required. Transketolase, we mentioned that it is an enzyme of HMP shunt. Why you should remember this? Because it is used to identify, you can say it is the marker. Activity of this enzyme is marker for deficiency of vitamin B1. We can use this activity to measure the deficiency of vitamin B1. Okay, let's go back. So in the fed or the fasting state, which pathways are regulated, which pathways down regulated, which enzyme is activated, which enzyme is inactivated, like that, the question will be asked. So remember, when I say fed state, this means increased glucose. And when I say increased glucose, this means increased insulin. This means increased insulin. So again, we come back to the same question. What are the action of insulin? In fasting, it will become the opposite. In fasting, it will become opposite. Decrease glucose, decrease insulin. So whatever insulin normally does, the opposite will happen. Third, insulin glucagon ratio. Very commonly, you will see the questions on insulin glucagon ratio. Insulin glucagon ratio is decreased. Which of the following enzyme will be upregulated? Insulin glucagon ratio is decreased. Which of the following pathways is downregulated? So, like that, the question will come. So, how to solve this question? We very frequently get confused when the question is there on the insulin glucagon ratio. So, let's see how to solve this question. So, we have the insulin glucagon ratio. Right, we have the insulin glucagon ratio. I'm sure all of you know that insulin and glucagon have a reciprocal relationship. If insulin rises, the glucagon will fall. If insulin falls, the glucagon will rise. Right. So what happens in the fed state? In the fed state, the insulin rises. When insulin rises, the glucagon will fall. When insulin rises, the glucagon will fall. So what is happening here? The numerator has increased, the denominator has decreased. What will happen to the overall value? The overall value will increase. Obviously, the numerator has expanded, the denominator has become smaller, the overall value will become more. In fasting state, the insulin will fall, the glucagon will rise. The glucagon will rise. What will happen to the ratio? What will happen to the ratio? Our numerator has become smaller, the denominator has become bigger, so the overall ratio will fall. Now this is very very interesting. Please look very carefully what I'm going to mention now. When insulin increases, the ratio increases. When insulin increases, the ratio increases. When insulin decreases, the ratio decreases. When insulin increases, the insulin glucagon ratio increases. When insulin decreases, the insulin glucagon ratio decreases. And if we know what is happening to the insulin, whether it is increasing or decreasing, immediately we can look at the table which tells us the action of insulin, and we can determine what is going to happen, which pathway will be upregulated, which pathway will be downregulated, which enzyme will be upregulated, which enzyme will be downregulated. In other words, when you get the question on insulin glucagon ratio, you don't need to read the term glucagon ratio, because the insulin glucagon ratio follows the trend of change in insulin. The change in ratio has the same trend as the change in insulin. And in the question, we are only supposed to talk about the trend, whether it is increasing or decreasing. We are not talking about the amount. We are only talking about whether the change is increase or the decrease. So again, the insulin glucagon ratio converted to the question action of insulin. The last question that will come across, which can be solved from here, is enzymes active in phosphorylated and dephosphorylated state. Enzymes active in phosphorylated and dephosphorylated state. For this, I'll give you a very simple mantra. It is applicable in majority of the conditions, about 99.9% cases. This will be applicable. What you have to remember: insulin causes dephosphorylation. What this means: if insulin is activating a pathway, the enzymes of that pathway will be active in the dephosphorylated form. If insulin is inactivating a pathway, the enzymes of that pathway will be inactive in the dephosphorylated form. In other words, they will be active in the phosphorylated form. So, just by knowing the action of insulin and by knowing that insulin causes dephosphorylation, we can quickly comment in which state the enzyme is going to be active. So, what we need to know, we need to know only one thing: what are the actions of insulin? And now we can remember the actions of insulin by another simple mantra. See, when does when does insulin increase? When does insulin increase? Insulin increases in to response to increased glucose. This implies the function of insulin. The function of insulin is to restore the balance. In other words, it is going to consume the glucose. All right. So insulin will try to consume the glucose. So insulin activates glucose consuming pathways. Now, when insulin is increased, this means already a lot of glucose is there, and therefore all the pathways which generate fuel, whether it is glucose, whether it is fatty acid, whether it is a ketone body, must be downregulated. They are not required because their efficiency is not as good as in glucose. Therefore, it downregulates pathways which are making metabolic fuels. Which are the metabolic fuels? You should remember the metabolic fuels. The metabolic fuels are glucose, fatty acid, and the ketone bodies. So this describes the action of insulin. This describes the action of insulin. Insulin increases the glucose consuming pathways and it decreases the pathway making metabolic fuels. So what we'll do, we'll quickly list this so that it becomes clear. Glycolysis, glucose will be consumed. Glycogenesis, glucose will be consumed. Lipogenesis, glucose will be consumed. HMP shunt, glucose will be consumed. Amino acid synthesis, glucose will be consumed. And cholesterol synthesis, also the glucose will be consumed. So insulin will increase all these pathways. All these pathways will be increased by the action of insulin. On the other hand, it will decrease the glycogenolysis from where we are getting the glucose. It will decrease the lipolysis from where we are getting the free fatty acid. It will again decrease the gluconeogenesis from where we are getting the free glucose, and it will decrease the ketogenesis. Now, one important thing to keep in mind here, the main fuel for gluconeogenesis, if you recall, are the amino acids. And when we are doing the gluconeogenesis, we are making use of the carbon skeleton, right? What happens to the amino group of the amino acid? What happens to the amino group of amino acid? The amino group will undergo conversion to urea by the urea cycle. So remember, the gluconeogenesis and urea cycle will be linked. If gluconeogenesis rises, the urea cycle will also rise. If gluconeogenesis is suppressed, the urea cycle will also be suppressed. They are linked. The gluconeogenesis and the urea cycle are linked. So you should remember this point. At this point, we should also quickly list the changes in analytes when we move from the fed state to the fasting state. What are the changes in the analytes when we move from fed state to the fasting state? I'll quickly draw a chart showing the changes in different analytes when we move from the fed state to the fasting state. We'll draw a table. Okay. On the x and y axis, we have the time and relative concentration. Time and relative concentration. Here, time zero is the fed state, meaning when the absorption from the gut, from the intestine is completed. When the absorption from the intestine is completed, that is time zero. So, what will happen after time zero? Because no more glucose is coming from the intestine, gradually the glucose level will start falling. The glucose level will start falling, but it plateaus at a certain level. It plateaus at a certain level. At the same time, when no more glucose is coming, glucose degradation is going on, the liver glycogen will try to support this glucose concentration. The liver glycogen will try to support this glucose concentration. This is the liver glycogen. The liver glycogen is typically exhausted in 12 to 24 hours. I don't know. Typically exhausted in 12 to 24 hours, an average of 18 hours. Okay. Now, glucose and insulin we had seen have a similar trajectory, meaning if glucose starts falling, the insulin level will also start falling. And we had also said that insulin and glucagon have a reciprocal relationship. So at the same time, the glucagon will start rising. At the same time, the glucagon will start rising. Now, insulin has a very strong inhibitory action on the breakdown of fatty acid because it inhibits the hormone sensitive lipase. We'll do this when we do the lipids. Insulin inhibits the hormone sensitive lipase. When insulin level starts falling, at that time, the free fatty acid level will start rising. The free fatty acid level will start rising. This free fatty acid will reach the liver where it will start getting converted into the ketone bodies. So first the free fatty acid has to be generated, then it has to reach the liver, at which point of time the ketone body synthesis will start. Please note, significant amount of ketone body overlaps with the exhaustion of liver glycogen. It overlaps with exhaustion of liver glycogen. So when we move from the fasting to starvation state, this conversion is determined by the presence or absence of liver glycogen. That is when we have the significant amount of ketone bodies. That is when we have the significant amount of the ketone bodies. All right, so this is the typical changes that we are going to see when we move from the fed state to the fasting and subsequently to the starvation state. Please note there is no graph showing the rate of gluconeogenesis. All right, the gluconeogenesis cannot be exactly determined. Exact value of gluconeogenesis cannot be determined because the change in glucose is affected by multiple factors. So the exact rate of gluconeogenesis cannot be determined. So it cannot be shown on the graph. Any questions about this graph? Any questions about this graph? Any questions about this graph? Anything you want to ask? The feed fast cycle is broadly divided into three, but sometimes we can divide into four stages. Sometimes we can also divide it into four stages. All right, we can divide into four stages. Okay, I'll describe again the free fatty acid and the ketone bodies. Actually, the free fatty acid ketone bodies will become more clear when we do the ketone body synthesis, but I'll do it again. See, the free fatty acid will be formed from the adipocytes. It will come from adipocytes. From adipocytes, it has to reach the liver, and in the liver, the ketogenesis occurs. Right, so when the free fatty acid reaches the liver, immediately ketogenesis does not occur. Initially the free fatty acid is metabolized to generate the energy. Once the ratio of NADH to NAD becomes in the favor of the ketone bodies, more and more free fatty acid are coming into the liver. That is when significant ketone body synthesis starts. So you will see the rise in the fatty acid and the rise in ketone bodies, they are quite some distant apart on the time scale. Okay, so ketone body starts rising late. The rise in ketone bodies corresponds to the depletion of liver glycogen. The rise in ketone bodies corresponds to the depletion of the liver glycogen, because as long as some amount of glucose is there in the liver, it can keep diverting the acetyl coenzyme A from fatty acids into the TCA cycle. Once the support from glucose is not there, the increasing amount of fatty acid cannot be diverted towards the TCA cycle. It is then converted into the ketone bodies. Okay, so these are two conceptual points to keep in mind. The rise of ketone bodies occurs much later after the rise of free fatty acid. The rise in ketone bodies overlaps the depletion of liver glycogen. Okay, let's go back. Okay, so fed state, fasting state, there are some tissues for which you should know this. First, for RBC, what is the fuel for RBC in these stages? Can anyone tell me the fuel for RBC in these stages? Anyone? Fuel for RBC in these stages. This one is very simple. You should be able to tell us. Anyone? Anyone? Fuel for RBC in these stages? No one? Very good. Glucose. Remember, in all stages, RBC are going to use only glucose. In all stages, the RBC are going to use only glucose. So the answer is glucose. Only glucose. The second one is brain. What about brain? What about brain? Anyone can tell me? What about brain? What about brain? Anyone can tell me? For brain, what is the fuel in fed, fasting, early starvation, prolonged starvation? We are talking about the fuel coming from the blood. We are talking about the fuel coming from the blood. For brain, what should be the answer? Fuel for brain. Fed state, glucose. Fasting, glucose. Early starvation, glucose. Switch has started towards ketone bodies. And prolonged starvation, mainly it is ketone bodies and little consumption of glucose. So remember, in prolonged starvation, the brain switches from glucose to ketone bodies. A lot of us make this mistake. We think that brain will always use the glucose. This is not correct. In prolonged starvation, the brain can switch to ketone bodies, but what you have to remember, you need to give time to brain to make this switch. If you suddenly decrease the level of blood glucose, then the brain cannot switch to ketone bodies. If this decrease in the glucose concentration, the fasting is prolonged, only then this switch can occur. Sudden drop in blood glucose will result in the loss of consciousness. Sudden drop in blood glucose will result in loss of consciousness. What about liver? What about liver? In fed state, in fed state, the liver will use glucose, amino acid. In the fasting state, in the fasting state, it will make use of free fatty acid, little bit of glucose, and amino acid. In starvation, free fatty acid and amino acid. Free fatty acid and amino acid. All right, free fatty acid and amino acid. What you have to remember, the liver, the liver cannot use ketone bodies. The liver cannot use the ketone bodies. All right. And the last one which you have to keep in mind is the muscle. Last one to keep in mind is the muscle. In the fed state, glucose. In fasting, free fatty acid. Early starvation, free fatty acid plus ketone bodies. In prolonged starvation, free fatty acid plus ketone bodies. As the ketone bodies will become available, the muscle can make use of the ketone bodies. Alright, it can start making use of the ketone bodies. Okay. Last in this list, last in this list is the kidney. Kidney. What about kidney? Glucose in the fed state. Glucose in the fed state. In a fasting state, it can make use of free fatty acid, but mostly it will make use of the amino acids. Early starvation, prolonged starvation, it will make use of the amino acids plus ketone bodies, amino acid plus ketone bodies. So kidney can make use of all glucose, free fatty acid, amino acid, and ketone bodies. Kidney can make use of all glucose, free fatty acid, amino acid, and the ketone bodies. All right. So keep that in mind. The liver cannot use ketone bodies. The brain cannot use free fatty acid. RBC can only use glucose. RBC can only use glucose. RBC can only use glucose. Muscles cannot efficiently metabolize the amino acid. I will not say that cannot use amino acid. They can use, but they cannot use it efficiently. They cannot use it efficiently. So this tells us about the fuel for the different tissues and different conditions, and it covers all sorts of questions related to the fuel for the tissues. We will stop here. We have a very small topic left, that is the galactose and fructose and associated disorders. We will cover them tomorrow, and we'll also be doing the questions related to the topics that we have done today on that tomorrow. Tomorrow, I think the session will begin early at about 4 o'clock. You'll be updated about the same by Dr. Manoj. So we'll meet again tomorrow at 4 o'clock. I'll wait for 2 minutes. If you have any questions, please feel free to send them across. Any questions? Any questions? The free fatty acid here can't cross the blood brain barrier. The ketone bodies cannot be used because enzyme for activation is absent. Any questions? The amino acid used by brain is not very efficient. It can use, but not very efficiently. It can use the amino acid just like the muscle, but the use is not very efficient. Pooja, can you clarify your question? Gluconeogenesis and urea cycle linkage. What do you mean by linkage here? Means are you talking about a reaction or a substrate which is common to both? Any other question? Okay, so if there are no more questions, I think we will stop here today, and the time for tomorrow will be conveyed to you by Dr. Manoj. So we'll meet again tomorrow during the class. Meghna, there is nothing to explain. You just have to remember that generally insulin causes dephosphorylation. Generally, the insulin causes dephosphorylation. What you can do is remember it like this. Okay. So you can remember it like this. The insulin increases the activity of these pathways. Therefore, the enzymes are active in dephosphorylated form. On the other hand, these pathways are downregulated, so are inactive in dephosphorylated form. In other words, they are active in phosphorylated form. Yes, the statement that insulin decreases gluconeogenesis and therefore urea cycle is correct. So because the urea cycle and the gluconeogenesis are linked, so if gluconeogenesis decreases, urea cycle will decrease. If gluconeogenesis increases, the urea cycle will increase. Okay, take care, bye bye, stay safe, and good night. We'll meet again tomorrow.